Good morning, everyone. I am Mosmi Biswas, student of T1 BSc Botany. Welcome you for the second day of National Seminar and Hands-on Training on Fascinating World of Fungi, organized by the Department of Botany under the DBT Star College Scheme in association with MycoAsia, Journal of Modern Mycology. Today, our first section will be by Dr. Damodar Shenoy, sir, Senior Scientist of NIO, Visakhapatnam, who has a vast knowledge and experience in the field of mycology. On behalf of Department of Botany, we would like to welcome sir with a bouquet of flowers. I now would like to call upon Dr. Pratibha ma'am to introduce our today's speaker, Dr. Damodar Shenoy, sir.
Thank you, Mosmi. It's my pleasure. Very good morning to all. It's my pleasure to introduce Dr. Damodar Shenoy, resource person for today's session. Dr. Belle D is a principal scientist at CSIR, National Institute of Oceanography, Regional Center, Visakhapatnam. Dr. Damodar did his MSc from Mangalore University. He has a certificate of teaching and learning from University of Hong Kong. He completed his PhD in 2007 with world-renowned mycologist Professor Kevin Hyde in the University of Hong Kong with specialization in microbial diversity, genomics, and molecular phylogeny. After a short stint as a postdoctoral fellow at Ottawa, Canada, Dr. Damodar joined CSIR Institute of Microbial Technology, Chandigarh in 2008. In 2013, he joined CSIR NIO Goa. Dr. Damodar has approximately 50 publications which are highly cited. They are in diverse fields like taxonomy of fusarium, coletotrichum, mangrove fungi, and marine bacteria such as bacterial diversity in oxygen-deficient waters. His recent research interest is on tar ball degradation by bacteria and fungi. You all know tar balls are causing enormous marine pollution. Dr. Damodar supervised 4 PhD theses and several postgraduate projects till date. Recently, in 2023, he has also acquired MBA from Mizoram University. Dr. Damodar is a founder of MycoAsia, online journal of modern mycology, and all its activities for promotion of mycology in Asia. His CV runs into pages. With a brief introduction, I welcome Dr. Damodar for this session. I hand over the session to you.
Good morning. Am I audible to all of you? The back, am I audible? First of all, thank you, Dr. Pratibha, for your kind introduction of me to the audience. I'm very happy to be in Dhempe College. Vedi, thank you very much for giving me this opportunity to the Dhempe College management. And I'm happy that we are MycoAsia is collaborating with Dhempe College, one of the oldest colleges of Goa. We feel very honored that we are collaborating with Dhempe College. Thank you very much for that. Thank you so much. Now, in this presentation, I want to talk about how to identify fungal species. I mean, is this question that important? First of all, so this question can be divided into three parts. First is what are fungi? Probably you already have got an idea after listening to last two lectures yesterday. Probably your teachers might have been teaching you about fungi during your BSc classes. I'm not sure about your syllabus now. Second thing is, how do we identify fungi? What is meant by identification? You know, is identification similar to taxonomy? Is identification different from classification? How identification is connected with the nomenclature? So there has to be some clarification or clarity on this topic, I believe. The important concept here is the concept of species. How do you recognize a group of fungi as a species? By just looking at them under microscope or just looking at them with naked eyes, like if you can see mushrooms, if they have similar features, probably you could group them as a species, under one species. Is it that simple? But considering today's audience, mostly the bachelor students, I'm not sure I'm willing to go to the deep depth of the taxonomy, but I would be giving some general ideas. This will be a hardcore taxonomy lecture. Before I go to the core of the presentation, I will give some interesting stuff about fungi, you know. So this is the plan. Next 60 minutes I want to spend talking about these things. First, what are fungi? Simple questions are already answered. Then I feel I should talk about some simple things about fungi. I'm going to share some 10 interesting facts about fungi. I mean, need not be facts, but my own set of information I want to share with you. I hope you will find that information very useful or interesting. Then I'll go to the core of the presentation, fungal taxonomy. Is fungal taxonomy just identification? What is fungal taxonomy? Is it a science? Is it a technique? What is fungal taxonomy? And how fungal taxonomy is different from library science? You know, in library also you classify books, right? But in fungal taxonomy also we group fungi in under different category. How taxonomy or biological classification is different from library science? Both are about grouping the things, or one is grouping books, another thing is grouping life forms like fungi. Then I wanted to share about my PhD work. Probably it is not required for today's audience. Anyway, last I will talk about what is a fungal species, how mycologists recognize fungal species. This is kind of complicated, but I'll try to make it a little bit easy for you guys. But before that, I wish to have a kind of interaction. You are free to ask me any questions in between. If you don't ask me, I will be compelled to ask questions to you. Then you will have to answer them also. Because 1 hour listening to the same person probably is not fun, I guess, especially about fungal taxonomy, especially on a Saturday morning in Goa. Anyway, so I wanted to talk about my basics of molecular phylogenetics. Probably I'll take up that in the afternoon before I demonstrate about mega. Okay. I hope everybody is fine in the online participation. Hope they can hear me. Yeah, great. Okay, 45 people are there. Very much for joining in for the online participants. Most of us are friends from MycoAsia, MycoIndia, and the Association of Fungal Biologists, and some of them are not probably associated with this platform. Thank you very much for joining in. I hope you are going to have fun listening to me. Of course, many of the things I have already talked in these platforms before, but this will be one more audience. Maybe there will be a different set of people here who would be keen to understand what I'm trying to say. So important question is identification. What is identification? In common our day-to-day life, how do we identify a person? Iska beta, iska beti, or iska ek, I mean, kale kale aankh, whatever, that you have some kind of preconceived information, or you have set of information about that person or a thing. Iska beta matlab there is already information uska father, right? Therefore, there is a priori information. Similarly, how does, when we are the largest, we have the highest number of people in the world in India, I guess, 1.4 billion people. And the government has a challenge of identifying all the people. I mean, if you cannot confuse one person with another, how do we do it? I mean, we use Aadhaar card, right? It's good if you have information about all the 1.4 billion people. Do we have all the information? If you don't have the information, suppose we have to go there and register. That means there is a priori information. Based on that only the identification can be done. If your biometric information is not there, then you will not be identified based on the Aadhaar, right? So that is the priori information. Similarly, how do we identify organisms like fungi? By just looking at them, the color of them, size of them, other features. Because fungi are not like they are not. They are very diverse in terms of their appearance, right? In fungi, you can have mushrooms. Yes, mushrooms belong to fungi. Mushrooms are nothing but fruiting bodies of fungi, right? And fungi are a separate kingdom. So we have mushrooms, macroscopic structures. We have yeast also fungi. We have molds, penicillium or aspergillus, all kind of things are there. So how do we identify these kind of things when they are diverse in terms of appearance? That's based on the already existing literature in the books or in different papers. And identification is an important problem. Based on the Google search, you can tell people are interested in identification of fungi. Either they might have gotten some allergy because of fungi present in their bathroom or somewhere on the wall, or they might have found some probably mushroom, beautiful mushroom in the garden. They are keen to know their, the name of the fungus. Not fungus, in that case, mushroom. They would ask if mushroom ka naam kya. Second question they will ask is being humans, man being man, is it edible? First of all, edibility is different from identification. So, but first step is very important. Identification is very important before we confirm the edibility of the mushroom. Therefore, the identification is an important step, and people are keen on understanding the identification of fungi for various reasons. So, how do you identify? You can say these belong to molds, they belong to mushrooms, they could be yeast. Is it that simple? You can identify a particular fungus to that kind of groups. Those are kind of very practical centric groups, for utilitarian, But in taxonomy, we have different ways of grouping fungi. You can group them under one species, and many species come under a genus, right? Many genera come under one family. Then we go up, we have at the last domain, right? That is taxonomy. So people are also interested in identification of the species. Before that, they should have an idea what is a species. As human beings, we are a separate species under genus Homo. Right, we are Homo sapiens. So that is the species of us actually. In case of fungi, how many species could be there? How many species of fungi could be there on this earth? Any idea? I mean, we have 1.4 billion people in India, probably 6 billion people in this world, right? 6-7 billion people. And how many fungal species could be there on this earth? 1 lakh? Any idea? No, probably we don't know about the Indian population. So how do we know about fungi? Never mind. So there could be, it is estimated there could be at least 15 lakh fungal species on this earth. Minimum. And the number, the guesstimate can go up to 1 crore. But we mycologists or taxonomists have so far described only 2 lakh species of them. What about the remaining at least 13 lakh fungal species? Where are they? Right? Therefore, identity of a fungal species is important. And when we don't have much, our research gap is quite large. In the sense, we know only about 8% of the 15 lakhs guesstimated fungal species. So around 8 to 10%. Remaining 90% of fungal species are waiting to be discovered. These novel species could be in this room, in the soil, or in your Miramar beach. If you can go further, maybe in the sediments of Arabian Sea. Or you can go to the deep sea, where Samir Damere can collect samples and identify fungi. That's how we are interested about fungi and their species and their identification. Are you getting my point? What I'm trying to say, that identification is an important concept, and it has got great practical importance. Classification can be a intellectual kind of thing, right? It could be a hypothesis driven, but the identification is very utilitarian. That means very practical. We do it in our day-to-day life. We do it in our mycology. We do it in plants also, right? People are interested in plant kya hai, ashwagandha hai, whatever, right? So I wanted to talk about. Okay, I got some 44 more minutes. Okay, 10 interesting things about fungi. Fungi are beautiful. Do you agree? Okay, at least after listening to last two lectures, probably you would say, okay, they're not that bad, beautiful, right? So here is a mushroom smoking here, right? Not really. Here is a mushroom actually releasing spores. As I told you, mushroom is a fruiting structure. The purpose of a fruiting structure is to make spores and release it in appropriate time according to the intellectual or intellect of that particular, whatever the level may be. So here are some more fungi. You said fungi are beautiful. Here you see the fungi are eating our food, destroying our food. Are they really that beautiful if they are destroying our food? Probably they are beautiful, but if you remove your own interests, they are still beautiful. If you remove human biases, they are still beautiful. Okay, and in Kannada they have saying. I'm sorry, I had to use this. In the sense, अपने बच्चे कैसे भी हो अच्छे ही हैं. So for a mycologist, fungi are beautiful only actually, because we deal with them every day. Therefore, fungi are beautiful, and we want to show the beautiful things about fungi to us. That is the purpose of this ceremony. Because fungi are perceived to be nuisance, cause lots of problems in the house. Fungus problem, they say fungus problem. जो भी है, whether fungi involved or not, is a fungus problem. That is the issue. That kind of villains have become these fungi. But we want to show some nice things about fungi. Of course, there are some bad guys out there. They are bad because of the circumstances they are living in. So here are some more beautiful mushrooms. These are from canva.com. If you use canva.com, you can get these photos. Here you find some fungi on culture medium, right? So, according to you, what is the largest living organism on this earth? Whales? You are doing botany, zoology, chemistry, right? You did not know about fungi, but you could know about animals. You could know about plants. Any idea largest living on the organism on this earth? Blue whale. Okay, almost there. But it used to be, according to the Guinness World Record, a fungus, a mushroom used to be the largest living organism on this earth. I mean, you must wonder, what is this guy telling you? ये blue whale देखो, ये mushroom इतना छोटा सा है, वो कैसे largest बन सकते हैं? तो mushroom के नीचे जो mycelial network है, that is also part of the mushroom, right? In particular case, the mycelial network of this particular mushroom, honey mushroom, the scientific name is Armillaria ostrae. Actually, it spreads almost in an area equivalent to 1,300 football stadium in one of the national park in the USA. And this mushroom is believed to be at least 3,000, 4,000 years old. That fungus has been, this mushroom has been living for last 2,000, 3,000 years in that forest in America. And because of the mycelial network, it was once considered the largest living organism on this earth till another organism actually replaced it recently in 2022. Still, that mushroom is the largest living fungus on this earth. The record is still there. I'm not telling it is world, Guinness World Record. These people are telling. So the fungi have got their own achievements in that sense. They have already, blue whales are nowhere actually. They have got better standing in terms of achievements. So if you know very simple things, life can be divided into bacteria, archaea, and eukarya. So where does fungi fit? Bacteria, archaea, maybe eukarya, because that is the last one. Actually, they belong to eukarya. Eukarya means eukaryotes, right? You have plants, you have animals, you have all kinds of life forms which have got true nucleus. Those are eukaryotes, yes. But you are studying fungi in botany department. I think they are still being taught in botany department, not in zoology. So are fungi a part of plant kingdom? Because you are studying in botany department, right? In the botany subject, you are giving exams in botany also, under botany. A small portion, mycology, right? Mycology, what is mycology? The study of fungi. I mean, probably I'll give more information about it later. Fungi are not plants. They are different because you must be knowing some features, right? Because our main difference is fungi are heterotrophic. Plants are autotrophic. Yes, that is the main difference. Other things are there. Of course, fungi have some relationship with animals in terms of evolution. Actually, fungi are closer to animals than to plants, according to the molecular data. I'll show the data later. But fungi are not animals also. Fungi are fungi. They have their own kingdom. That is kingdom fungi. So this was the data I was talking about. According to this paper, animals and fungi are each other's closest relatives. Here is the tree of life. You can see mushroom here. That is the lineage of fungi. And you have animals after that. And you plants have somewhere at the below, right? I think I can use this here. Here you have plants. Oops, sorry. I think I'm okay. Anyway. And as you know, fungi, as I told you, fungi can be microscopic, they can be macroscopic, right? They are diverse in their forms. And if you consider the ancient history of the earth, who came onto the land first? Plants, right? Okay, according to some evidence, actually it was the fungi who came onto the land first, from where maybe an aquatic system like sea. And it is kind of suggested there were large giant mushrooms as tall as 8 meters. So I am almost 1.8 meter, right? So imagine 8 meter tall mushrooms. So the giant mushroom present in the ancient world, even before the plants could come on the land. So they not only they were present, they dominated the ancient world, it seems. Okay, that's about some fun, interesting stuff. But what happened later? Where do you find giant mushrooms now? At least 1.8 meter tall. I'm not sure actually. You can find them horizontally and probably not on vertically. How many fungi? I already told there could be 1.5, and we have so far described 2 lakh fungal species. And people ask me whether a mushroom is edible or not. There are around 14,000 mushroom species have already been described. Out of them, 2,000 species are proven to be edible out of 14,000. Remaining, we don't know whether we can eat or we can just spit out. It's up to you. Okay, and here you find some 18 popular ones, edible mushrooms. Why I'm showing you? Because mushroom market is not a small market. It's not like it's not as big as spongy market. In the sense, it is larger in terms of money. The mushroom market is around 50 billion US dollars. That is the market size of the mushroom market. That means you can make money if you can grow mushrooms. You can eat some, but remaining you can sell. Right? I think Madam was telling you are getting trained in mushroom cultivation. Focus on that. You might be a successful entrepreneur in mushroom cultivation. Not only selling mushroom, you can sell mushroom-based products also. Tomorrow you can make leather material out from mushroom skin actually. So here you find 18 different kind of things. I mean, fungi are notorious for their diseases in plant. Therefore, they have got bad names as villains. But they not only, I mean, they cause some problems in human beings also. But not all fungi are problematic. Many of them are very collaborative. You know about arbuscular mycorrhizal fungi. What do? They actually collaborate with the plant root system and help in the nutrient exchange and communication. You have lichens. Here is the association between of a couple of fungi and algae or cyanobacteria. And these lichens are known to be an indicator of climate change. Yes. And you know about penicillin. That was the first antibiotic of the world. I mean, in a modern scientific way. And that is from a fungal culture of Penicillium rubens. The fungal origin is Penicillium rubens. This is how the fungus looks. Here at the top, you can see the fungal culture. Then here are the micromorphological character. Here is a phylogenetic tree. Any idea what is a phylogenetic tree? A phylogenetic tree is just a representation of evolutionary relationships of a group of organisms. Here you have a group of organisms belonging to the genus Penicillium. Here you have many species belonging to Penicillium, and here at the top you have the sequence information of Fleming's penicillin producing culture actually, and it is clustering with other members of rubens, Penicillium rubens. This is how we identify. So I'm coming to the point, how do we identify now? Not just based on the morphology, sometimes you have to use more than morphology. So what you see is morphology. And what you don't see, but you can sequence it and later on see is molecular information, right? Molecular information, nothing but DNA information, DNA sequence data. Penicillin is not the only thing. We have so many other molecules we got from fungi. The future is all about, probably according to me, future is about drug-resistant molecules to treat depression. And these molecules derived from magic mushrooms. What are magic mushrooms? These are mushrooms which can produce psychedelic compounds. And these what are psychedelic compounds? They modulate the neuro system, and they will have soothing effects on our brain. It will make you calm down. So that will be very useful in treating depression. Not just depression, drug-resistant depression itself. So these psychedelics from magic mushroom has got great potential in future, and American investors are investing money on this. It is expected that we can that we would have this kind of molecules in next 10-15 years. Lots of interesting things, except if we are hit by an asteroid, only fungi can save us, because fungi don't need sunlight and they can get nutrients from the soil, and you can consume mushrooms if you are not able to grow any other plants. This is how this article argues. In case of an asteroid attack, it cannot be an attack. When they are bombarded by an asteroid, the fungi can be our saviors. This is what this article argues actually. So this is about the fun part. Now let's go to the more difficult part, the taxonomy. So I got some 30 more minutes to teach you something about fungal taxonomy. What is taxonomy? It's the science. What is the beauty of science? You can always question science. Yes? How do you question science? With better evidence, a better set of tools and better evidence. Taxonomy is not a technique, it is a science. Why do I say? When I say science, it is hypothesis specific. And a taxonomy has, fungal taxonomy, for example, you can have 3 different things under taxonomy. First one is classification. What is classification? That is the hierarchical grouping of living organisms, living organisms. Here you can have species at the bottom and you go up to the domain in life. In between you have genus, family, order, class. Am I fast? Are you able to pick up? Fine, right? Fine, okay. So I'm just telling you, important aspect of taxonomy is classification. That is more intellectual thing. Whether here we discuss, can we group this particular set of organisms, set of fungi under genus or one species or a genus? I mean, so this is how we intellectually discuss, and that intellectualism is quite interesting. It can have all kinds of effects. I will explain that later if you can comprehend that. Second thing is based on the classification system, based on the information we gather during in generating a classification system, you can identify. Identify is a binary process, right? Yes or no. In Aadhaar card, you give a fingerprint, it will say Badam Belle Damodar Shenoy or no record found. That's it. It's the binary process. Same with fungus. Oh, this fungus has these features. If you compare with the database, not really, there is no unique database for all the fungi. You have to dig into literature and find out and compare with the type material, what you call type material or the reference material. If these characters match, oh, okay, this fungus is this particular species. If the characters do not match with any of the known fungi, then what you do? Then you create a new category for this fungus. That is describing new species. And you cannot describe new species giving Mera fungus, Mera Ganga fungus. No, you have to give proper names. Aspergillus. So what is Aspergillus? Find out what is Aspergillus. Niger is black, right? So similarly, you can give names, but there are some instances where mycologists are very enthusiastic to respect their teacher. They give names under teachers, genus name for respecting the teacher. Natarajaniya, I guess. He was probably not a teacher, but one of the legends of it. So I am told there are some fungi which has got species name of the girlfriends of mycologists. So my girlfriend helped me in the sample collection and I'm inspired. So I think there are a couple of those crazy things happen, not very routine, but I'm just trying to make some humor out of taxonomy and nomenclature. Okay, so what are the three components of taxonomy? Anyone? I'm just waking you up from your sleep. Classification, identification, and nomenclature. Brilliant. So who is the father of biological classification? Carolus Linnaeus, right? So because of him, we have got my binomial nomenclature. Or a biological name should have two names, genus name and species name. So we still continue that. So here you have a photograph of his book, Systema Naturae. And traditionally, fungi were grouped under plant. Traditionally, I'm talking about 1973. Here you find fungi as a subkingdom in plant kingdom. And you had Myxomycota and Eumycota. So because of recent developments of in the era of nucleic acid data, especially after 1978. In 1978, what happened? Carl Woese actually divided the life into three domains. What are those? Archaea, bacteria, eukarya, based on ribosomal RNA information. So this is a famous paper, got him the Nobel Prize, I guess. Carl Woese. So this development had implication in taxonomy also. Now you can use molecular data to classify organism, and when the sequencing became very easy comparatively, so we started developing primers to amplify gene region. So here you have a famous paper by White et al. Mycologists cite this paper regularly. This paper actually describes primers. Primers are nothing but these are small fragments of DNA which helps in the amplification of a gene region. For example, ITS region. What is an ITS region? Never mind. Before that, let me tell you, this is the recent classification of fungi based on many gene regions and other data also, biochemical data, morphological data. This is by Hibbert et al. So what is special about it? You know, you might have studied Ascomycota, Basidiomycota, Zygomycota, Chaetota. In this classification, there is no Zygomycota. What happened? Zygomycota was found to be polyphyletic. Matlab, it is not a natural group. It doesn't represent a natural grouping. What is natural grouping? Which is similar, which is happening in the nature, based on our perception, based on our hypothesis. So Zygomycota is not there. I'm not sure whether you are teaching these things to your students now. Teachers, not the Zygomycota is not there, and the NCERT people will probably update mycology portion, please. because this is the latest information that kingdom fungi has been divided into different things, but not zygomycota. Chytids are used in a restricted manner. Chytidomycota. And we have microsporidia, which used to be in animals. Now it's a part of kingdom fungi. This is all because of the revision based on molecular data. That is the power of molecular data. Oh, now barcoding. When you go to supermarket, you have what you have. You get so many things. If you start checking everything, you will take half an hour for one customer. So what do you call barcodes? So you have barcode scanner. So here is an idea. Can we identify organism based on a short fragment of DNA sequence? For example, in case of fungi, you have a region called ITS region. So this region is around 500 bases long actually. Can we use this, sequence this and identify? It's a good idea. It will simplify many things. You don't have to spend years and years learning morphology. You can just culture the fungus and send it to Bangalore and pay 2,000 rupees, get the ITS sequence information, compare with the known information in the Gen Bank. But is it that easy? Do we have sequences for all the fungi described so far? No. Do you have information from all the type materials? No. It's an ambitious thing to think about it, but to practice it is not easy. We are facing lots of problems, but it is one primary barcode region for kingdom fungi. Okay? That is the ITS region. Please remember this ITS region. Other things you forget, no problem. I'm not going to talk about nomenclature. These are my PhD work. I said I'm not going to talk about it. I got some 20 more minutes, maybe. Yeah. Huh? Yeah, I'll just talk for next 20 minutes. So important question is, what is a species? What is a species? Anyone? We are Homo sapiens. Why all the human beings, are put under Homo sapiens? That's because we create taxonomy. Therefore, we are put under one category, right? There could be some reasons, right? We compared to the fossil data. I don't know. There could be one. There is only one species in Homo, right? Genus Homo. I don't think there is any other species at the moment. Conceptually, species are segments of evolutionary lineages that have evolved independently from another. This is a very simple, but according to me, very effective definition of a species. Species are segments of evolutionary lineages that have evolved independently. Evolutionary independence is the thing. That means a group of individuals, a group of fungi have separated from the closest relatives in terms of their genetic material. So these a group of fungi cannot have reproductive processes with another closely related fungi. Therefore, this becomes two different species. It's a good concept, right? It's very simply, in a simple manner, we have explained, defined what are species, what is species. But in day-to-day, how do you practice it? This is a species concept. Therefore, to practice in day-to-day life or practice our research, we have something called species recognition. Species recognition criteria. These are nothing but practical tools by which you can recognize a group of fungi as fungal species. So you can use morphological character. You can look at them, external appearances. How do they look? How do mushrooms look? Whether they have same size of the cap, whether they are of same color. Well, that about when you can see it without microscope. When you see with using microscope, you can have to just have a greater details, like whether the spores are of same shape, whether of the same color. How about the spore producing structures? Are they same? It gets complicated, But you can use morphology to recognize a group of fungi under one species. That is nothing but morphological species recognition. But recently, because of DNA sequence data, you can sequence DNA information, DNA from these fungi, and you can reconstruct their evolutionary relationships, and you can identify lineages from their hypothetical recent ancestors. that becomes phylogenetic species recognition. This here how it becomes a little bit complicated, especially for the bachelor's level student. So last one is you can use, if you use one gene region, that actually tells the history of that gene, not necessarily the history of that species. I'm not sure I can make it more simple than this, but basically in practical life, in day-to-day life, mycologists can recognize fungi using morphology, DNA information, that is phylogenetic species recognition. When you use multiple gene regions, that becomes very complicated concept called genealogical concordance phylogenetic species recognition. Basically, you are using gene regions which share similar evolutionary history. So are you already overwhelmed? Okay, that's the fun, I guess. That is the aim of my presentation, to make everybody overwhelmed with taxonomy. Anyway, so any questions? Just ask me a couple of questions before I go to more serious topics. Anyway, morphological species recognitions. Let me give this one example. Coltotrichum. I'm not sure many of you have heard about this genus. Very important fungal genus. Here is a very famous species, Coltotrichum gliosporidis. Very not easy to pronounce also. But so this is how the fungus looks on the culture plate. No distinct morpho characters actually, or morphological features. This is how these are the conidia. Here I got aprosorium and all. So previously, any fungi which looked like this were classified under Coltotrichum gliosporidis. And this particular name was associated with around 900 plant species as pathogens or endophytes or probably saprophytes of soil or something. Basically, this morphospecies was widely reported because of its different roles as endophyte. In 900 plant species, imagine, belonging to 450 plant genera. So this is how, in a very simple manner, they recognize a group of fungi to be belonging to the species gliosporidis under the genus Coltotrichum. That was the morphological species recognition. But what happens when you sequence? What happened when we sequenced? When you do identification, when you do taxonomic analysis, you have to do comparison. You cannot compare with anything what is available in the world. You have to compare with something called type material, a reference material. When somebody describes a new species or a taxon, you have to identify one particular plant material, herbarium material, or culture as the reference material. Here, when you do reorganize the taxonomy, you have to use the type materials or type sequences from the type material. What happened when they use the sequences from the type material? There was problem with the type and typification. I'm not going to get into that. When they use the type material and the sequences from them, they found that, so these are the lineages, these are the clades, the group of sequences. Those are called clades. They found that the type material is clustering with some group of fungi, but the similar looking fungi are all over the tree. Basically, it means this is polyphyletic. In phylogenetic analysis, we call it polyphyletic. A group of fungi which may look similar, but they are from different ancestries. That is poly. And as per the Darwinian thing, you cannot have a taxon which is polyphyletic. So you have to redefine it. You have to give new names. I'm trying to make it very simple. Basically, after molecular phylogenetic analysis, we got more names for the similar looking fungi based on the DNA sequence info. Here is the example. This is the Coltotrichum gliosporidis, which is defined in a very restricted manner in using molecular data of the type material. This is not just one gene region. I think there are 4 to 6 gene regions. And for under one morphospecies, 23 phylogenetic species were identified because that morphospecies was found to be polyphyletic, not representative of any natural grouping. Is it fun? We are destroying taxonomy here, actually. Not destroying, we are dismantling what is existing, and we are coming up with something new, 23 new names. You ask any plant pathology, do you think they will remember all these names? They will still remember Coltotrichum gliosporidis, sensulato. taxonomist कुछ भी कर लो, we will use it Coltotrechum gliosporides, Sporiadis, Sensuato, जो 1890s में जो describe किए वही definition में accept करेंगे. But science has evolved. Taxonomy might have been kind of rattled, but the science has improved. We have got better understanding of species boundaries under this morphospecies, under as 23 phylogenetic species. Our PhD student worked on one of the morphological similar Coltotrechum siamens, which is phylogenetically different from Coltotrechum gliosporides. We found the genetic diversity, then we described new species. Then what we found that there are some tools called coalescent methods. So when you do phylogenetic analysis, you got branches on a phylogenetic tree. So these are small branches. So based on that, you give names. There is no guarantee that the reproductive isolation is confirmed. You know, what is reproductive isolation? Evolutionary independence is confirmed because of this through reproductive isolation. Like in animals, if two set of organisms stop interbreeding, then it is reproductive isolation, right? Similarly, on a phylogenetic tree, we may have branches separated. Doesn't mean we have evidence for the reproductive isolation. But when we do coalescent methods, it provides the information about the reproductive isolation. So what we thought 7 species under Coltotrechum siamens, now it has been synonymized. Now we have only 1 species. Remember, 1 morphospecies, 23 species. Now 7 species goes back to 7 species, 1 species. So this kind of, there are 2 groups in taxonomy, groups of taxonomies. It's funny actually. Some people want to split, some people want to clump. This will go on till taxonomies, they are surviving on this world. That's the bread and butter of taxonomies. That's how we want to improve the science. It's a perception based on the evidence. Therefore, I always ask some of my friends, do you believe in fungal species? See, 1 species become 23 species because of molecular data. Afterwards, because of refined data set, 7 species were merged into 1 species. It's this kind of, what you call transition, or this kind of instability we'll continue to have. We'll continue to have. Probably this is through this kind of instability, this kind of upheavals, this kind of developments, which may not be very comfortable, because taxonomy has to have some stability, right? You cannot change names every 5 years, every 10 years. But now, so far we have done what morphology, uske baad, single gene, uske baad, multi-gene, abhi uske baad, the genealogical concordance, uske baad, abhi coalescent methods. Now, future is all about artificial intelligence, machine learning, predictive mycology, predictive taxonomy, and all this linear type material, all this typification. I'm not sure linear system of hierarchical system will be there. Probably our understanding of taxonomy will change, our understanding of fungal evolution will change when you get all kinds of data, omics data. When I say omics, genomics, proteomics, Dr. Samir will talk about proteomics after. Probably he might have left NIO now to reach here. But basically, future is about lots of data, big data, what you call big data, mining in our genomes, not our fungal genomes in case of mycology, proteomics data, transcriptomic data, secondary metabolite profiles. And that's what I'm coming to. So you will have in future molecular data, morphological data, ecological data, analyzed using sophisticated bioinformatics and modeling approach. When we use modeling, we are going to have artificial intelligence and machine learning in future. And we will have integrated taxonomy. And we're going to test every taxon based on eco-evolutionary theory. I said taxonomy is a science because every taxonomic category is a hypothesis. And based on the evidence, it can be proved or disproved. That is the beauty of science. What is the difference between library science and taxonomy? In taxonomy, we have incorporated Darwinian theories of evolution actually. So we believe in the monophyletic lineages. I thought I should stop here, and this presentation I'll give in the afternoon before I do the practicals. Ashish. I hope they are okay.
So, last question.
Yes, please. Yeah.
First, what is ITS and why only ITS? Or why only those regions? Why not all the anything? A lot of genome research.
Yeah, I mean, that's a good question. I was a part of this project, barcoding project actually. See this DNA barcoding, where is it? This DNA barcoding concept was applied, I mean, was attempted for the butterflies and fishes in eukaryotes. In eukaryotes, you don't use ITS. We use, I mean, for eukaryotes, we use cytochrome oxidase 1, might have heard COX-1 or CO1. So there was an ambitious idea that all life forms should have only one kind of barcode. But when we tried to do it for fungi, there were many problems. One was that multiple copies of ITS in the same genome. The second thing is that ITS could not identify many of them satisfactorily. I mean, cytochrome, sorry, I'm going back to cytochrome example. Cytochrome oxidase could not identify, was found to be ineffective in fungi because of many regions or many of the multiple copies of it. Then they consider many ITS also, they consider ADNS also. ITS has got optimal kind of evolutionary rate, which can have aid in the species identification. The rate of evolution is quite good enough for the rate, species identification. But in many cases, in case of Poltertrachum, ITS is ineffective. In case of Fusarium, it is ineffective. In case of Penicillium, also it is ineffective. It cannot identify the species accurately. If you want to differentiate Aspergillus niger and brasiliensis, ITS will not work. You will have to use beta-tubulin and calmodulin. So these mycologists, as a consensus, chose ITS region. That's the primary barcode region. You could always have a secondary barcode region. Okay. Yeah. Thanks for that question. We will be answering that in the afternoon because I thought of continuing with these. See, very complicated slides are here. Wait. Here. Where I'm going to talk about bootstrap and other stuff. What I'm thinking in the afternoon, I'll spend 20 minutes for these slides. Probably people are already overwhelmed with too much of taxonomy. I don't want to with the more complicated phylogenetic concept. No, I don't want to burden them with so much of concepts. Probably they will run away from taxonomy. But idea is we'll spend 15-20 minutes in the afternoon talking about this concept. Then then you can download the software and we'll have fresh these concepts fresh in your mind on your mind. Then when you do the practicals, you can ask the questions. Whether 70% boost. First of all, what is bootstrapping? Bootstrapping is a very general word. Bootstrapping. It has I'll tell explain it later. This is a statistical way of finding out the support for this separation of branches. If two branches have got a bootstrap values of 50%, out of 100 times, 50% of the times you can find this kind of branching. But remaining 50% of the time, it can go wrong. It cannot be the same. Is it a good value? We expect 100% to be the best one because there cannot be 101%. 100% would be the best bootstrap value, but you may not always get 100% because gene regions may not have phylogenetic signal. One gene region may not have. You need to use multiple gene regions to improve the bootstrap for the analysis and all. But normally they say 70% to theek hai. So there is no science behind it. 70% acha hai, 69 acha nahi hai. It's very subjective. Normally बोलते हैं कि 70% ठीक है, acceptable. It's all about statistics, probability. 70% ठीक है, 30% टाइम इधर उधर जा सकता है, तो ओके चलो एक्सेप्ट कर लेंगे. That's how it is. Ideally, we should have more than 90%. Statistically speaking, 95% जो होता है ना आपका p value or whatever value. And we have better things to do in terms of statistical analysis. We have Bayesian values in addition to bootstrapping. But for every tree should have bootstrap values. Without any bootstrap value, the tree is it may be a beautiful tree, but the bootstrap values are not presented. All the beauty is useless. I told you when they analyzed the sequences, they found these members of zygomycete, traditional zygomycete, are clustering all over, all the tree, different groups. They were found to be, they have multiple ancestors.
They have to be modified.